Imagine you're looking at a young animal. Everything about it looks normal, but inside its cells, something has started to look old. It's mitochondria, the tiny structures responsible for producing much of the cell's energy, are becoming fragmented and dysfunctional. And the strange part is that researchers didn't have to wait for the animal to age. They changed one biological pathway, and the mitochondria started developing characteristics normally associated with aging. Then, they did something even more interesting. They changed the pathway back, and some of those aging-related changes improved. So what exactly did they change? The molecule at the center of this story is called phosphatidylcholine. It's not a futuristic drug. It's not a mysterious compound found in some exotic plant. It's a molecule your body already makes and uses as an important component of cellular membranes. And researchers are finding evidence that its metabolism changes as organisms age, which raises a fascinating question. Could changes in something as basic as a cell membrane actually influence how mitochondria age? To investigate that possibility, researchers turned to one of the workhorses of aging research, Caenorhabditis elegans, tiny roundworms. They may look nothing like us, but they're extraordinarily useful for studying aging because they have a short lifespan and many biological processes that scientists can manipulate experimentally. And this gave researchers something they can't easily do in humans. They could deliberately disrupt a specific biological pathway. And watch what happened. The researchers interfered with genes involved in producing phosphatidylcholine. And something unexpected happened. In young worms, the mitochondria began developing characteristics normally associated with aging. Instead of maintaining healthy dynamic networks, the mitochondrial structures became more fragmented. That's important because mitochondria aren't supposed to be static little batteries. They're constantly changing. They connect, they divide, they move, they adapt to the demands of the cell. And aging can interfere with that flexibility. But the researchers had a much more interesting question. If disrupting this pathway made young mitochondria look older, what would happen if they restored it? So they tested phosphatidylcholine itself. They also tested choline, which cells can use to produce phosphatidylcholine. And the result? Some of the mitochondrial abnormalities improved. The mitochondrial network became healthier, and aspects of mitochondrial function were restored. Now, stop here for a moment, because this does not mean scientists reversed aging. But it does tell us something potentially important. It suggests that at least some features associated with cellular aging may be biologically adjustable. That's very different from saying, scientists found a cure for aging. They didn't. The experiment was performed in experimental organisms, and the effects were specific to certain mitochondrial changes. But scientifically, there's an important distinction here. There's a difference between observing that something changes with age and discovering a mechanism that can actually move that change in the opposite direction. And that's where this story gets interesting. We've all heard mitochondria described as the powerhouses of the cell. That's useful, but incomplete. Imagine instead that each cell is a city. Mitochondria are part of the city's power infrastructure. But the power stations aren't isolated. They're constantly adapting to demand. They can merge, they can split, they can redistribute resources, they respond to stress, and their physical structure matters. Think about an electrical grid. If everything is connected and flexible, power can be redirected when demand changes. But if the grid becomes fragmented, individual sections may still generate electricity, but the entire system becomes less adaptable. Something similar can happen with mitochondrial networks. During aging, mitochondria can become more fragmented and less efficient. So researchers aren't simply asking, are mitochondria getting worse? They're asking, what is causing them to change? And that's where phosphatidylcholine enters the story. The name sounds complicated, but its role is easier to understand. Phosphatidylcholine is an important component of biological membranes. And membranes aren't just passive walls. They're dynamic structures. They have physical properties that influence how cellular machinery works. Mitochondria depend heavily on those membranes. So if the ingredients making those membranes change, the mitochondria themselves may be affected. According to the research you're covering, phosphatidylcholine production declines as organisms age, and that decline is associated with changes in mitochondrial structure and function. But association isn't the same thing as causation. That's why the worm experiment matters. Researchers weren't satisfied with simply observing that the molecule changed. They interfered with the pathway. And when they disrupted it in young animals, the mitochondria started behaving in ways associated with aging. That makes the story considerably more interesting, because then they ask the opposite question. If less of this pathway pushes mitochondria toward an older state, can restoring it push them back? This is one of the most important parts of the experiment. The researchers supplied phosphatidylcholine and tested choline, and some of the mitochondrial abnormalities improved. The system wasn't magically transformed into a young animal, but specific aging-related mitochondrial features moved in a healthier direction. That's the kind of result that gives researchers a new mechanism to investigate. There's a huge limitation. A worm isn't a human, and a laboratory experiment isn't a longevity treatment. We can't take a result like this and immediately conclude, take phosphatidylcholine and you'll slow aging. That's not what the experiment showed. It showed something much narrower. A particular metabolic pathway appears to influence aspects of mitochondrial biology in an experimental model. And that gives scientists a question worth pursuing. But now comes the part that makes this story more relevant to us. Does anything similar happen in humans? Researchers didn't stop with worms. They also examined human biological data and conducted experiments using human cells. And they found evidence that phosphatidylcholine metabolism is connected with biological changes associated with aging in humans as well. That's interesting. But we need to be extremely precise about what that means. Human data can show us patterns. Human cells can tell us how individual cells respond. Neither one automatically tells us what happens when you change the pathway inside an entire living person. And then researchers noticed something else. The pattern wasn't necessarily identical in males and females. In women, changes involving phosphatidylcholine metabolism appeared particularly noticeable around menopause. Now, this does not mean phosphatidylcholine causes menopause, and it doesn't mean taking phosphatidylcholine treats menopause. That's not what the evidence establishes. But the observation raises an intriguing biological question. Why does this metabolic system appear to change during such an important transition in aging? And this points to something we sometimes forget. There isn't one biological process called aging. Aging is more like dozens, or potentially hundreds, of systems changing at the same time. DNA damage, inflammation, immune function, protein quality control, metabolism, cellular senescence, hormonal changes, and mitochondrial function. So phosphatidylcholine isn't the aging switch. At most, it may be one adjustable component inside a much larger network. Imagine your biology as an enormous control panel. One dial influences inflammation. Another affects glucose metabolism. Another influences cellular repair. Another affects mitochondrial function. Most of these controls don't operate independently. They interact. So changing one can potentially help one system while affecting another. That's why the exciting part of this research isn't necessarily the molecule itself. It's the possibility that some aspects of aging may be controlled by specific biological states. And biological states can sometimes be changed. But there's still a mystery. Why does phosphatidylcholine production decline with age in the first place? Is it simply a consequence of aging? Or does that decline contribute to the mitochondrial problems associated with aging? Those are very different possibilities. If the decline is merely a symptom, restoring it might have limited effects. But if the decline contributes to the problem, then it could become a much more interesting therapeutic target. And right now, researchers don't have that answer. One possibility is a biological feedback loop. Aging changes metabolism. Those metabolic changes alter mitochondrial membranes. The mitochondria become less adaptable. Cellular energy management becomes more difficult. And that creates additional stress. If such a cycle exists, then intervening at one point could potentially change what happens downstream. But again, that's a hypothesis. And this is exactly where we need to separate interesting biology from proven human therapy. And this is probably the question many viewers are now asking. So, should I take phosphatidylcholine? Based on this research alone, there isn't enough evidence to say that you should take it to slow aging. The study does not establish an anti-aging dose. It doesn't prove supplementation extends human lifespan, and it doesn't show that taking more phosphatidylcholine will make your mitochondria younger. This is an extremely important distinction. Here's why. Your body isn't a bucket that simply needs more of every useful molecule. It's a highly regulated system. A molecule can be essential at one concentration and have very different effects at another. And a pathway that looks beneficial when manipulated inside a cell may behave very differently when you change it throughout an entire organism. That's why promising molecular biology can take years to become useful medicine. First, you need to understand the mechanism, then safety, then dosage, then long-term effects, then human clinical outcomes. So let's strip away the hype. What do we actually have? We have evidence that phosphatidylcholine is an important component of cellular membranes. We have evidence that its metabolism changes with age. We have experimental evidence that disrupting the pathway can produce aging-related mitochondrial changes in worms. And restoring the pathway can improve some of those changes. We also have human biological data suggesting that related metabolic changes occur during aging. That's fascinating, but it's not yet a longevity treatment. And honestly, the most interesting part may not be phosphatidylcholine at all. It's what this experiment tells us about the way researchers think about aging. For a long time, it's tempting to imagine aging as a clock. Year after year, damage accumulates. Systems gradually deteriorate, and eventually they fail. But experiments like this introduce another possibility. Some age-related changes may involve systems that are actively regulated, and regulated systems can potentially be manipulated. Now, that doesn't mean you need an experimental molecule. In fact, while scientists investigate these pathways, some of the strongest tools for maintaining healthy aging are remarkably ordinary. Movement, strength training, good nutrition, adequate sleep, not smoking, and maintaining cardiovascular and metabolic health. None of those sounds as exciting as a hidden molecular switch. But biology doesn't care whether an intervention makes a good thumbnail. Take muscle. As people age, losing substantial muscle can affect much more than appearance. It can affect balance, strength, mobility, recovery, the ability to climb stairs, the ability to get up from a chair, and ultimately independence. That's why longevity isn't simply about adding more birthdays. It's about preserving the biological capacity to live those years well. And physical activity gives your cells a reason to adapt. When you move, your muscles demand energy. Your cells respond. Energy production has to increase. Mitochondrial systems have to adapt to repeated demands. That's one reason exercise research is so important in longevity. You're not trying to activate one magical molecule. You're repeatedly challenging an entire biological system to remain functional. The boring truth is actually remarkably powerful. Move regularly, maintain muscle, eat a nutrient-rich diet, get adequate protein, sleep sufficiently, don't smoke. keep blood pressure, blood sugar, and cardiovascular health under control. These things aren't a shortcut to immortality, but they influence many of the systems that determine how well your body functions as you age. And unlike an experimental molecular pathway, we already have decades of human evidence behind many of these behaviors. But now imagine where this kind of research could eventually lead. What if doctors could monitor biological aging at the level of individual systems? Instead of simply asking, "How old are you?" they might someday ask, "How healthy is your mitochondrial function? How resilient is your metabolism? How well are your cells maintaining protein quality? How much inflammation is present? How quickly are these systems changing?" That would be a very different way of thinking about medicine. And the future probably won't be one magic anti-aging pill. Aging is too complicated for that. It may eventually involve combinations of interventions. One targeting inflammation, another protecting muscle, another improving metabolic health, another affecting cellular repair, and perhaps another influencing mitochondrial function. But we're nowhere near knowing whether that strategy will actually work. The phosphatidylcholine pathway is one clue, not the final answer. So let's go back to the beginning. Scientists did not discover a cure for aging. They did something more scientifically interesting. They changed a specific metabolic pathway in young organisms, and some features of their mitochondria began resembling those seen during aging. Then they restored the pathway, and some of those changes improved. That's not immortality. It's not proof that humans can reverse aging. But it does challenge a simple assumption, that every change associated with aging is necessarily irreversible. And that may be the question worth remembering. Not, did scientists find the anti-aging molecule? They didn't. The better question is, how many parts of aging are actually adjustable? Maybe only a few. Maybe many more than we currently realize. We don't know yet. But every time researchers identify a biological system that changes with age, and then discover that part of that system can be pushed in another direction, they give us another piece of the puzzle. And perhaps one day, the science of aging won't be about finding one switch. It will be about learning which switches matter, when to adjust them, and how to do it safely. That's the real story behind this discovery. If you want to follow that research as scientists figure out what these discoveries actually mean for humans, subscribe to Longevity Explained. Because the most interesting question in longevity may not be how long humans can live. It may be, how much of aging can biology actually change?